Good morning, everyone. Welcome to the Hindu Physicians International Distinguished Lecture Series. My name is Dr. Adityanji, and you see the logo and the name of the Hindu Physicians International. Our Mool Mantra or motto is Shariramadhyam Khalu Dharma Sadhanam. That actually gives the Swasti mantra. And when we say Sharira Madhyam Khalu Dharma Sadhanam, we are talking about body being your temple. And that is true for our patients, but also for the physicians, because most of the time physicians neglect their own health. So body is your temple to worship, and first duty for everyone is for their own body. So a little bit about what is this lecture about. To be on the record, today is August 23rd, Sunday, 2026, 10 a.m. Eastern Time. Our distinguished speaker is Dr. Pareek, and he will be giving us a lecture on From COVID-19 Immune Memory to Cancer Treatment, Approach to Personalized Cancer Vaccines. And before I hand over the platform to him, a little bit about Hindu Physicians International. Why did we need this? HPI is a faith-based charity by physicians of dharmic origin to serve the entire humanity. It has a self-governing board of directors with 5 members. It is a non-profit organization registered in the state of New York. Our self-governing board has 5 members, and we focus on Sanatan concepts like Naraseva, Narayan Seva, Vasudhaiva Kutumbakam. We are here to serve the humanity, but under a Sanatan framework. That's why we decided to create this faith-based charitable institution. We serve the humanity irrespective of race, ethnicity, religion, and nationality. But of course, under Sanatan platform. I will not go into the more details. We are also looking into other faith-based medical charities, and there are plenty. There were none as far as dharmic physicians were concerned. So we thought that there is a time and space for doing selfless service under a Sanatan framework. So the next slide will tell you about who are on the board of directors. I'm not going to go through the names. We have an international advisory board. Again, I will not review the names. These people are chosen for their competence. There's an executive committee in which we invite younger physicians, medical students to do day-to-day work. We are always looking for physician volunteers for taking responsibilities for this organization. Next, please. What are our objectives? Community service or seva activities worldwide. Our long-term objectives are to develop HIDER, humanitarian assistance and disaster relief teams, rapid response medical teams, focus on prevention of environmental degradation, and promote prevention, research, and access to treatment for common illnesses. Membership is free. You go onto the website, there's a form, Google form, you fill it up, whether you are a physician, surgeon, whether you are a respiratory therapist, social worker, psychologist, medical student, resident, membership is free for everyone. And there's a community seva project. If you are doing a community seva project, you just fill up the form for getting HPI certification. We don't take the ownership. You remain the director of the project, but it is done under the banner of HPI. So we are now coming to the distinguished physician lecture series. And the next slide will give you the flyer that you people saw to log into this particular event with Dr. Pareek's picture as well as his bio. Let's move on to the next slide, please. Dr. Pareek is an accomplished biomedical researcher and a successful bio-entrepreneur based in Cleveland, Ohio. He received his biomedical public health and business training at premier research institutions, including Indian Institute of Science in Bengaluru, National Institute of Health in Maryland, USA, and Case Western Reserve University in Cleveland, Ohio. He has authored numerous scientific papers, peer reviewed book chapters, review articles, published in peer reviewed biomedical research journals. His breakthrough scientific discoveries have earned him numerous national and international awards. Currently, he is working as an adjunct professor at Case Western Reserve School of Medicine and Mendel School of Applied Social Scientists at CWRU. Next, please. He is also a scholar of global health issues and healthcare policy management and works with an international NGO. He is co-founder of Celarum Inc., a US-based multinational biotechnology startup focusing on developing therapies against cancer and immune disorders. Dr. Pareek's insightful academic and business vision, as well as impeccable social and public political relationships in India and USA, have enabled several collaboration ventures among the two nations. He is basically now focusing on translation research from basic science to clinical science. He is co-inventor of certain, therapeutic things. So at HPI, we have focus on Seva. We have done a lot of Seva activities, and something that is going to come fairly soon on September 5th and 6th is a Sanatan Sportsathon 2026. We are trying to encourage youth of the Sanatan community to take care of their health, reduce the screen time. So we decided to promote this sports event September 5th and 6th. Please join it, please participate in it. Again, we go on to, what is Sharira Madhyam Khalu Dharma Sadhanam. We participated in International Yoga Day. We were one of the sponsors for that in Cleveland. And we have done numerous Seva activities, supported the BAPS Walkathon charitable event. That's a certification from them. So I will not go into rest of the slides, which give numerous local Seva projects at various places. But without further ado, it is my honor and pleasure to invite Dr. Tej Pareek. But before I do that, if we can go on to the last slide, which is Swasti Vachan. Dr. Siva. Om Swasti na Indro Vrudrashavaha Swasti na Pusha Vishwavedaha Swasti nastaksho arishtanemihi Swasti no brahspatirdhadhatu. Om Shanti Shanti Om. So the platform is your, Dr. Tej Pareek. Please go ahead. Pleasure to have you on this platform.
Namaste, Adityan ji, Shiv Kumar ji, and all curious audience who joined this talk today, which was published recently last month, late week of last month, and got a lot of media attention. It is just because the landscape of cancer treatment is very rapidly evolving and changing. And science is always, as we say, it is scientists will never believe what the truth is because truth is constantly evolving. So we are seeking truth, and our that curiosity in general to understand science gives us new solution. As a scientist, what I used to learn 10 years back has completely changed. Not changed, I would say that evolved, and evolved in a succinct manner where we are very close, if I can say, to have some possible cancer treatments, which may not be as painful and as deleterious as it is at today's time. So the question which I will start the talk with, and I will give you some remarks. I talked with Adityanji. I just thought that I will keep this talk as simple as possible. The complexity is published in the paper, but I would be happy to answer any questions if any audience has at this time. But idea is to just introduce to what cancer is, which all of us may be knowing. Maybe we will just revisit that. And once we do that, then also just deep dive into where this cancer vaccine space is evolving. So the fundamental question, which scientists are curious and asking for a very long period of time, and that history is it goes very back. And the question is, can we train our immune system to fight cancer? As you all may be knowing, that immune system itself is quite robust in our body, and nature has given us that system to fight the outside pathogens or any kind of abnormalities which our body face and correct it in timely manner. So the idea is this: can we utilize it to fight cancer as well as that immune system is able to fight with a lot of other ailments and diseases? Before I start, I just want to disclose my conflict of interest. I serve as Saloram's board of director, and I am also a stockholder in the company. I am also co-author of this manuscript and co-inventor of this technology. And I just want to disclose that Protexi technology presented here today is fully owned and commercialized by company Saloram. And just because I have this participation with Saloram, I just want to be clear, this today's talk is in no way an advice to any medical treatment or any investment advice. So just take it as your educational curiosity purpose. Uh, I would also just to set up the landscape, what we will cover in this talk and what we will not. So just we are clear, we will talk a little bit about how cancer develops, what today's treatment landscapes are, what is this buzzword cancer vaccine, which is just like now in the media, and then this paper and other discoveries by Moderna and Merck has brought to again to the limelight there. And what is the published preclinical research of Protexi, if there is? What this talk will not cover, just to be clear, it is not going to give any kind of a diagnosis to any cancer patient or recommending any treatment to any patient or replacing any current guidelines of cancer treatment. That remains just to the the patient's discussions with their physicians and the treatment plan which they are going on. I also want to disclose that the current Protexi paper published in Nature Communication, it is just an investigational cancer vaccine. Its safety and benefits in patients yet to be tested, and we are opening a clinical trial here at University Hospital in Cleveland, first time for the sarcoma patients. So just to give you an idea, and you can all know about it and might have heard or read about it if you are more curious about it, that cancer is not today's word. This goes like thousands of years back, even in Egyptian mummies, there has been signs of cancers. So the human body experiencing cancer is not a modern day disease, which sometimes people confuse with. It is just like now we know the diagnosis of it, and now we know how to cure it. And a lot of experiments has been done since that time. Patients used to be removed, their cancer used to be taken out. And just to give you an idea, there was a lot of experiments done long time back where people, when they have the tumors, they will infect these patients with some kind of a viral infection and then see whether some kind of a response is there. And then, surprising enough, some patients will be cured. The reason was not known. That time, science was not evolved, but these experiments has also been done. Just to give you the breadth of this disease and how severe that is and what we want to achieve and why this should be a priority of all researchers and scientists and doctors, and this should be the focus of our population as well, to understand what this disease is. Uh, just as the data of 2024, there are about 20.6 million new cases diagnosed worldwide. Just take these number in your mind, how big these numbers are. About 9.8 million people die globally every year because of this disease, and about 34.4 million new cases are projected worldwide by 2050. So there is no stop or pause on this disease. It is constantly increasing. And if you think about it, about 1 in 5 people will develop cancer. And again, I just want to make sure it is developing cancer. We are not projecting how many people will die with cancer. But quite a bit chance is this, that look at the number on the Zoom call today and think about it, like 1 in 5 people will develop cancer in their lifetime. And that the number is skewed a little bit more towards women, as 1 in 9 men and 1 in 13 women, towards the man, 1 in 13 women will die from it. And there are reasons for it. Just to give you the statistics for the US alone, US cancer diagnosis will exceed more than 2 million just this year. So just in 2026, the data suggests that we will have about 2.11 million new cancer cases diagnosed just in United States. That comes to about 626,000 deaths by cancer just in America. And if you simplify the math, that comes to about 1,700 deaths per day. That means that by the time this talk ends, we will lose 60 lives, 1.2 deaths every minute. So it's a very deadly disease. We do not know much about it. We know a lot about it, but we have not resolved the complete mystery, why it happens, how it happens. But there is a huge investment of science from US government as well as across the globe, resource-wise as well as talent-wise, has been invested into this research of cancer. And you know that the cancer is not once the simple thing. There are several different type of cancers. We say there are about more than 200 types of cancer, which are diagnosed based on the location of that cancer. There are about different 60 organs where this cancer is located, and that's how it is defined. Few cancers which one of us might have listened, and this is a disease. I Whenever I go and give a public talk, I can say that uh, there's a very big chance that someone in our family might have had the cancer, or at least we may be knowing someone who might have suffered with cancer. And out of all the cancer cases which we hear, see, learn about, prostate cancer, breast cancer, lung cancer, and colorectal cancer are on the top of it and contributes to about 50% of cancer related death, the numbers which I shared earlier. Now, there is also, I just thought that this is an interesting statistical public health data. You will see that the most new cases which are identified of cancer, that is prostate cancer, breast cancer, lung cancer, and colorectal cancer. But it doesn't mean that the more number of cancer cases caused the cancer related death also, although the prostate cancer number is the highest number, but usually it happens at the advanced age, 50 plus. It also happens at early age, but the most cancer related death still happens with the lung cancer, that contributes to about 125,000 deaths per year. Colorectal come on the second, pancreas come on the third number, and then the breast cancer come on at the last. The reason for that being that the nature of cancer in each organ, each tissue is different. The some cancers are more metastatic, more aggressive, and also there are more treatments available for some of the cancers. Like breast cancer, it used to be quite deadly 2 decades back, but a lot of advancement and happened to understand the nature of this specific tumor. Uh, I also wanted to give a relief to, we all have to appreciate the effort of investment and resources contributed through our scientific community for years. Quite often, we develop this perception that cancer is on rise, everyone is dying with cancer. Once cancer is declared, then death is inevitable. However, since 1991 till today, there has been about 34% decline in cancer-related death. We have saved about 4.8 million extra lives, which would have been lost what the science and development was there in 1991. And now today, about 18.6 million people live in US even after diagnosed with cancer. So there has been a lot of growth. One question whenever I share this statistics is that why 1991 was the peak of the cancer, and there was obvious reason. A decade before, there was a fashion of smoking became part of the culture, and lot many deaths were there in 1991 because of that. Lung cancer cases were maturing and eventually resulted in death. And at that time, science was not that evolved as well. If you think about 1991 is not that far the timeline. But after that, a lot of diagnosis and other cases, other strategies are also evolved that led to reduction in the death. So every science count, everything which scientists publish that lead to some kind of enhancement of knowledge. Uh, I will just put this slide because in a simplistic manner, what the cancer is and then how it happens, all of you may be knowing, but just for simplicity purpose, I call it unrestrained drama of immortal cell causing mortality. So in our body, in a simplistic way, if I can call, every cell has an expiry date. And that cell is constantly, most of the part of our body cells are constantly dying. And then new cells are made by repurposing, reutilizing the biological material of that cell. So this recycling of our cells are constantly happening. That means that new cells will be formed and old cells supposed to be discarded. Now, there are several reasons because of that. These some of the cells decide not to die. And in a simplistic way, I can say they sometimes forget to die, and sometimes they have gone through so many environmental pressures where they learn to live longer. And either of the case, whatever that case is, and that happens eventually result into genetic mutations or several other reasons. That genetic mutations might have inherited in that cell or might have acquired due to environmental pressures or the lifestyle which that person is going through. That cell decide not to die. Just to give you an example, like there is a bigger trend in America of tanning. If you see, I think it is about like 5 or 6 years back, Surgeon General report where he actually asked and did a call, public health call, to ask to make the skin cancer the largest pandemic of our time. And the reason has been that people are utilizing this tanning practices, overexposure to sunlight, and these things can create mutations to the cell. I will not go into the detailed biology, but whenever a cell decide not to die, that's a declaration of the person is moving towards the path of the mortality. So it's an unrestrained drama of immortal cells causing mortality. And most of the time the patient do not we do not lose patients because of that tumor. If that tumor remains localized, that can be excised from the tumor site. But mostly these tumors learn because there is a real estate problem in that tumor, and then some of the cells decide to escape from that tumor and utilize the blood capillaries, lymph node, and other spaces and invade into other tissues. So a cell which is in nature, it was a liver or it was lung, it decides to metastasize and reach to other organs. And that becomes the major cause of the death. So we say that the cancer diagnosis tumor is one part, but most of the cancer related death happens because of the metastasis of the cancer. And I would also just put this slide because cancer is not just one disease. It is evolution inside the body, which is constantly happen. So, as I said, a cell controls the break, and that break can happen because of the genetic and cellular safeguards stop working in that cell. And then once that break stops, that cell is decide to continuously expand. And then once they start to expand, they become a different sort of cell in our body, and then they learn and adopt to fight with our immune system. They learn to create resources for that continuous growth, and they are out of control of our inner bodily mechanism. And then once that metastasis happen, then this disease spreads, and then things are out of control. Again, we accumulate these mutations throughout our lifetime. Just to give you an example, cancer doesn't happen that if incidents happen today and the person will expose to cancer tomorrow. If a person is smoking, for an example, it may take 10 years, 20 years, who knows what amount of time for him to develop this lung cancer. If person is exposed just for the skin cancer melanoma purpose, Shiv Kumar ji was asking me before the talk, if a person is having a 5 times sunburn in his lifetime, there is a 90% possibility that after the age of 20, he or she will develop skin cancer at some point. So this takes years for these mutations to reflect in body and eventually turn into cancer. So they can happen because of genetic mutations which are acquired or accumulated, or the exposure to the environment, or these people are we are predisposed to some kind of a variants or chronic inflammation in our body that can create that situation. So how do we deal with it? What we are doing today and where science is evolving. So cancer treatment targets disease in different ways. And currently there are many options. One option is obviously surgery. A surgeon will go remove a localized tumor. There are a lot of great imaging technologies through which you can maximize the visualization of the tumor, but still there are not the absolute technology which can see everything. These cancer tumors are notorious. They hide. They are such a minute size, and then they stop replicating. So that can allow them to escape from this imaging. But still, if you can image it, the surgeon can remove the localized tumor, and most probably that will solve the problem. But that also create several other problems, because that tumor can come back if it is not excised permanently. And then sometimes physicians will use radiation, and radiation exposure, that localized radiation exposure to the tumor, will create genetic mutations into that. It will create the DNA damage, and eventually that tumor will be melted out, or the burned out, if you will call it. The problem with radiation is this, that it also affect the neighboring tissues, and the treatment, because of the radiation, the genetic damage which happened in the neighboring tissues can create several other problems. And who knows that genetic mutations can also be cause of other tumor development. Then the patients also go through chemotherapy, and sometimes combination of radiation and chemotherapy. And chemotherapy are the exposure of some agents systematically, and then these agents do not differentiate in cancer cell or the normal cells. So their nature is to kill any cell which is constantly dividing in the body. So when cell is dividing, their DNA is fragile, and these chemotherapies just go and kill the cells. So the problem is this, that they may kill the tumor, but they may also kill the normal cells. And that's why whenever you see that this treatment has increased survival rate at this point, usually we are talking about the 5 years of survival rate. So this radiation and chemotherapy, a lot of advancement have happened. It can protect the person from the spread of the cancer. However, the quality of life is significantly compromised, and it is not guaranteed that these treatments will absolutely cure the treatment. Now there are a lot of targeted therapies which have evolved in past 10, 20 years, which block specific proteins which are known by researchers now. And then the last one, which I will be focusing on, is immunotherapy, where you somehow stimulate the immune attack on the tumor, or somehow you remove the brakes off the from the immune system so it can exponentially grow inside the tumor and eliminate the tumor from the body. Just before go that, I will just give a 1 minute idea of what the immune system is, how it works. There are a lot of learned person here on the talk, but I will just, for the simplicity, I will say that the first protection of defense, if we have in our body, that is our skin, the mucus, the which help us from the protective chemicals from any kind of threat from outside. So that is our first barrier of protection. And then we have in our body a innate response and adaptive response. We call it innate immune system or adaptive immune system. And as the name suggests, the innate immune system are the cells which already know how these foreign pathogens look like. They have the immediate attack to that, and that includes our macrophages, phagocytes, natural killer cells. So as soon any sort of infection will come, they will attack, they will eliminate. It is rapid. But life is not simple, and then these threats can be enormous, unknown. So that's where we have this adaptive immune system, which is consisted of something called T cell and B cell. Just remember that what they are. B cells create antibodies against that antigen, and then T cell are actual executors of eliminating that that infection from our body. And during this process, our immune system also develop memory against these antigens. So in future, if suppose that pathogen or that threat comes again, our immune system remain prepared, and then they can eliminate that from our body. So this is important to understand how it works. Now I will just tell you these three cells which we have to focus on the technology and what we are doing here at Cellorem. We are focusing on one specific cell called dendritic cell, which is part of the innate immune system. It actually talk with both innate and adaptive immune system, and then T cells, which take the knowledge from dendritic cells. So any pathogen which comes to the body, these dendritic cells come in contact with that. They take the information of that foreign pathogen. They identify abnormal proteins from that, which are infectious. We call them antigens, and then go and show them to T cells. So just take it for a simple analogy. A chief commander of an army will understand what the how the enemy looks like, and he will decode that information to the army on the ground and ask them to go and attack find the invader, and then eliminate it from the from the society, if you will call it, if it is an enemy. So that's how our immune system works. So dendritic cells, you can consider them as the chief commander cells, and T cells you can consider as the executor cells, which will actually fight with the pathogen and remove it from that. Now, there are also checkpoint signals which can switch an immune system response before the tumor is cleared. So, what happens that there are on tumors, it's kind of they create a break. So, immune system which are capable of eliminating any kind of tumor from the body, there are some breaks created by these tumors just to survive more, and then immune system cannot enter into the tumor, and that's why these tumors continue to grow. So scientists are also trying to develop therapies which can remove the break. And if you would have heard the recent advancement of Moderna cancer vaccine, it is a combination of their mRNA vaccine with Merck Keytruda, which is a PD-1 PD-L1 inhibitor, which is the actually the removing the break. That's what they are doing. And also the idea is to somehow, once these tumors are, we call it cold tumors, and cold tumors we call because there is no space for immune cells to enter inside the tumor. So the idea is to see if somehow we can create an environment where these immune cells, like this army, can enter into the tumor, and then it can become a hot tumor and can, the war can be in a fair way where this immune system is engaging with cancer cells. So the prime question which we ask, and several other researchers are asking, can we help the immune system to see, attack, and remember cancer? So the how we are going to achieve it. So first we have to release the brake. We have to supply better fighters. In this case, T cells. Somehow we have to prepare them for this fight, and then we also have to teach this army the right targets. So we have to identify unique antigens from the cancer cells and then show it to T cells. There are misnomer or misunderstanding, I would call, as we call them, cancer vaccine. Our general understanding of past 100 years is vaccine is something which you give in advance, and cancer vaccines are generally preventive. But in space of cancer vaccines where we are developing them, there are both kind of cancer vaccines. One are preventive cancer vaccines, and you all might have heard of HPV vaccine, hepatitis B vaccines. They reduce the risk of cancer because the antigens are known. You can give the body in advance and then develop memory against them. And then there are therapeutic vaccine, which we are developing at Saloram, and companies like Moderna, other companies are also in that space. They are developing. It's called therapeutic vaccine, where you, once the incidence of cancer happens, then we go uniquely identify the antigens from that specific patient and develop a vaccine specifically for that patient. So mostly these therapeutic vaccines are personalized vaccine, and that is the limitation of these cancers. These are very cost cumbersome research activities. And if you can, I just showed you that how many million people are suffering with cancer, and if we have to design cancer vaccine to each patient, is it possible economically as well as the logistic wise, and what is the cost involved in that? But right now, including Protexi, including the Moderna vaccine, which you might have heard about a couple of days back, these all are personalized cancer vaccines, because each person cancer is different. We may call it a prostate cancer, but prostate cancer A persons and prostate cancers B person is completely different. And even within the tumor, there is so much heterogeneity of the prostate cancer of person himself, that you cannot create a treatment for all the prostate cancer cell, even in that single person. So there are different kind of cancer vaccines are developing. Dendritic cell cancer vaccines, which we are developing, called Protexi. mRNA neoantigen vaccines, which are developed by Moderna. There are also other people are developing peptide protein-based vaccines. And then there are vectors and other cells are also utilized to develop these cancer vaccines. The difference in mRNA, peptide protein, and dendritic cell vaccine is some which I just want to highlight here. If a company or an investigator can develop mRNA or peptide-based vaccine, it is easy to inject into patients. A patient will go, and just like you get a flu vaccine, doctor will inject it in your arm, and then you are done with it. Maybe with some adjuvant, which the side effect will go away after some time. However, these vaccines, if you inject mRNA and peptide, at least the current science suggests that their effectiveness is not as much as you will desire, because immune system do not respond immediately to that localized mRNA peptide injectable sites, and then response is not that great. And that's where the space of dendritic cell cancer vaccine was evolved. And in this process, patient has to be admitted to hospital. Through leukapheresis, their blood will be collected, then dendritic cells will be collected. Then the patient tumor biopsies will be utilized to identify tumor specific antigens. And then these antigens will be exposed to the dendritic cell, and then these patient cell will be implanted into that patient. So it's a cumbersome process, but their effectiveness is superior than this mRNA and peptide vaccine. As you all know, that there is a landmark in the US of this Provenge, which is an FDA-approved autologous cellular immunotherapy for certain patients with metastatic prostate cancer who are resistant to castration option for available for these prostate cancer. So field is evolving. Already these vaccines are getting approvals in the market. uh, why we have this therapeutic cancer vaccines been difficult so far, even though we are doing this research for past 10 years, because we know the weak targets. We do not know the exact targets of the tumor just because of their heterogeneity. There is insufficient help. Once we put these, activate these T cell inside the body, the army is not enough to fight with the enormous number of enemies inside the patient body. As I said, there are cold tumors. Their immune system cannot invade into that, and then also the body learn how to escape from the immune system. So I will just enter into what this protaxi is. And in that, I will tell you there are two different cells of T cells, which I talked about. One is called CD8 killer cells. So these are the actual black cat commandos, if you will call them, which will go and can engage with cancer cells. So these are the cells which will actually go and fight with cancer cells. But there are other cell types which are not involved, and their job is not to fight with cancer, but we call them helper cells, and these are called CD4 cells. So what people are doing, and we were also doing in our company for a long period of time, where we were engaging and boosting the CD8 killer cells through the dendritic cells, this blue color representation inside the cell, and we were able to achieve the tumor regression. However, it was not that robust. So what we are doing now, that these DC dendritic cells are capable of engaging with both CD8 cells and then CD4 cells. So can we develop a mechanism through which we can engage both of these cells together? So just imagine something like this, that where these the real attackers, the black cat commandos, can also help with the local army, and can we attack them more robustly to eliminate that tumor from the body. So if we can create such environment where both these CD8 killer cells and CD4 helper cells can come together, and then we can remove the brake from the tumor, let the tumor let the immune system enter inside the tumor, and can that will be helpful for that or not. And we are utilizing the chief commander cells, dendritic cells, to create that option. And how we will do it, so we are utilizing something called COVID memory. And what we realize, we use a lot of antigens in our body which are our immune system have memory against, and we realize that COVID memory is more is an easy approach to develop these vaccines, because globally most of the human beings either develop this memory either through vaccines or the natural infection in their body. And in order to do that, we first we create a platform technology where these dendritic cell will recall helper memory once we inject these COVID vaccines. So CD4 cells will get activated, and then we create this put also the tumor antigens in that patient. And once they both come together, the response is robust, and they can remove the tumor. So this is a simplified diagram for the what this Protexi cancer vaccine which we are developing at Salorem. And if you can walk with me on this diagram, a patient will come, his peripheral blood will be collected. We will be doing two sequencing, one of his normal blood as well as his tumors, which are excised from the patient to identify the specific antigens. From this blood, we will isolate the monocytes through specific cultures in a clean clinical environment. We will convert them into dendritic cells. And once these dendritic cells are ready, we will load these dendritic cells with two set of antigens. One are which are part of the COVID protein, SARS-CoV-2, and also the specific antigens which are specific to that person's tumor. So once these two COVID peptides and the tumor antigens are loaded, this will be our final product, which we call Protexi. And now this Protexi will be injected about 5 million cells back to the patient. And then this whole process can be completed in 10 days. The limitations of this process, as you can imagine, it needs a clinical lab nearby the hospital where all this process can be done. And also these patients who are there, They go through a lot of treatments continuously. So the antigens which we identified 10 days before may rapidly change 10 days after. But what we figured out that the smaller is the time frame, the better the response is. So this is the paper. I will not go into science, and maybe I will try to wrap up in another 5 minutes. Adityanji, if you will allow me.
Sure.
Uh, this asks a couple of questions. We have mouse tumor models for melanoma and breast cancer. That is, typical preclinical models are used in research before testing any therapy into humans. We tested the mechanism of CD4 dependence, immune memory, and epitope spreading. We also try combination therapy with anti-PD-1, which the same Moderna research clinical trial results came few days back. And also we tried our partner company, Medpacto, who is developing TGF-beta 1 receptor vectrosertib uh, in combination with that treatment, and that made the treatment more effective in these preclinical model. We also did one advancement, if you will call it. It's called humanized mouse model because mouse do not get COVID. So we have to create a mouse model where we replaced that mouse immune system with human immune system of a COVID memory driven human peripheral blood. So that created an environment where the immune system was of human, but the body was of mouse. And then we tested this vaccine, and results were quite supportive. That gave us a lot of confidence that this can be safe and effective in humans, but the trial need to be still done. And then once we do that, this immune cold tumor, as I said, this red color cells depicted here, that turned into immune engaged, or you will call it immune hot tumor, where these red, blue, green color immune cells were invaded into the immune system and tumor was eliminated. This is a couple of data. You can go into paper and read more about it. But essentially, you will see, just look at the bar where Protexi is written. The tumor burden was significantly reduced than the conventional peptide injections or the antigen injection itself. In case of this melanoma, you see this red line. The tumor was completely eliminated within 3 months of the time frame. And one interesting thing which we did, that once we these immunized mouse, if we take their immune system and then replaced in another mouse which has not seen the tumor, and challenge that mouse with the tumor, uh, interestingly, that mouse which adoptively received the immune memory from another mouse was unable to develop tumor, which suggests that this immune response developed in the mouse is stable and that can prolong. We did another experiment that in this mouse, after 98 days, when tumor was completely removed, we injected another tumor in that mouse to see whether this memory is sustainable. And then this mouse did not develop the tumor again. What it suggests that at least in these preclinical models, this immune memory can be sustainable and quite effective uh, in eliminating the tumor. And then this one is with that breaks which are there on the immune cancer cells, which block immune cells to enter inside the cell. So this anti-PD-1, Shiv Kumar ji, you were asking about Keytruda, that antibody we used to remove the break along with our Protexi. And you can see the response was quite robust, and tumors were more aggressively removed in the body. And then this right side is another vactosertib, which is a cytokine TGF-beta 1 inhibitor. Once we use that, then the response was also high. So what we believe that Protexi itself is a quite effective vaccine approach. And if we can combine it with other therapies, which can also support in eliminating these immune checkpoints, or can support in creating a robust immune response, that can be more effective approach. And this is the results which I was telling you, that humanized mouse which we created and then injected with the COVID peptides to improve the health of the CD4 cells. The results were remarkable, and then tumors were shrink, as you can see here in the green line in this graph. Um, so what is supported by this paper and what is not yet established, I think I have to clarify that. there is a plausible antiviral memory mechanism which can be exploited to target the tumor, which is non-tumor specific. But if we can create a tumor specific immune response along with the antiviral immune response, that combination can be a useful approach to eliminate the tumors more effectively. There is a dependence on CD4 helper T cells, which is a new knowledge. Earlier, we used to think that only CD8 killer cells are involved in that. And then there is a tumor control we established in several preclinical models. In this paper, breast cancer, melanoma, and now we are advancing into sarcoma. That data is also there. And then there is a feasibility with vaccinated donor human immune cells that we tested in the humanized mouse. What is not yet established, I just want to make sure the safety in patients has not been tested. We are applying for FDA approval to test it first time in humans here in University Hospital. That clinical efficacy in humans need to be tested. Breast cancer dose, schedule, or combination, which will be the that need yet to be optimized. And then how broadly the benefit will translate, we do not know. There are a lot of research which are successful in mouse, has failed in humans, but we remain optimistic as scientists, and we are very eager to try this technology into humans and then see whether we can make a difference. The next step is carefully designing first in human study, which is important for safety, feasibility, immune activity, and early signals. Once that safety study is done in phase 1 trial, we will be moving to phase 2 and phase 3 if FDA asks us to go that big. So I would like to just end that future may not be one universal cancer vaccine as the field is evolving. That is today's understanding. Who knows what tomorrow will be? But looks like that we have to move from a fit for all approach in this cancer treatment field to the personalized cancer treatment, which are created and curated based on the nature of that person's specific tumor. And more, we will see now that there will be personalized immune response will be utilized to eliminate this deadly disorder. The business landscape will also change because different kind of labs will be needed, different kind of expertise will be needed to deliver these therapy. I just want to acknowledge the team which put their hard work on this work. The team at Saloram, there are investigators at University Hospital and Case Western Reserve University who were part of this team, and they are the team behind developing this treatment, developing countless hours, not giving up, and then thinking about this idea and believing in this when no one was thinking or believing about it. So the credit goes to them. So thank you very much. I will take any questions for next 10 minutes or till when Adityanji will allow me.
Thank you very much indeed, Dr. Tej Pareek, for a very fascinating lecture and making very complex science simple to understand, because we have a mixed audience right now with people who do not have medical background and people who do have medical background. A wonderful lecture and a wonderful opening for future treatments of cancer. So we are open for questions. Since it's a small audience, people can just turn their uh, video on and can ask question directly. But please be precise. Don't give a lecture to Dr. Tej Pareek. Just ask a simple question. Please go ahead. Whoever wants to ask a question. Wow. Okay, Professor Anup Kumar, you have a question?
Yeah, I thought I will get it going. So, yeah. So, Dr. Tej Parikh, how much time does it take to when something basic scientific research has been done, you have tested it on a mouse, and now taking it from there to making it really an FDA approved drug? Could you speak about how much time usually takes and how much money is required to even get it implemented?
Yeah, thank you for asking that, Anupji. And being in the research field, scientists have done research on that as well. This question as well. What is the uh, average time frame and the money which is required, and data is quite staggering. It takes about, understanding is this, that on average 14 years for a drug to come to the market in shelf available for patients from the time of discovery in a laboratory. So once the proof of principle studies are done, from that time to come to the market, it takes about 14 years. There is first the safety, the preclinical evidence has to be established, then safety studies in humans, then the larger population, then mixed population, and making sure there is no adverse effect. And we all have to give credit to, it looks like quite cumbersome, but FDA is quite robust in making sure before the treatment safety of humans are ensured. And that's why this time takes. There is also data which suggests about 1.5 to 2 billion dollar, billion for B, goes in one medicine discovery. So you can imagine about 14 years, about $2 billion. That is usually taken up by any drug before it comes to the market. So it's a rarely a scientist will see a discovery which he did on bench in his lifetime before his retirement, he would see in market. I would also give you one another data if that is not enough. About 99% of research ideas which are driven by enthusiasm, hypothesis, and all, about 99% of them will fail. And they can fail after 1.4 billion is already expended, or they can fail after first $10 has expended. So it's a quite risky, quite time
Mhm.
cumbersome approach, but whole society contribute to this research together, and they all hold behind it. The dollars come from federal government, which is contributed through public money. It also comes from private equity, where people would like to promote and invest into this research. So I hope I answer your question.
Yeah, I
So I
can you
have,
just
oh,
yeah, go ahead, please. Shiv Kumar, please. Kumaran, please go ahead.
yeah. No, no, if you've completed your discussion, I wanted to ask a question.
I just wanted to follow up if can AI speed up this RCT process, the randomized control trial process?
Not that clinical trial, I do not see that has improved. However, the AI has completely revolutionized the field, at least what I see. Drug discovery process has been accelerated
Okay.
in enormous way. The companies who are doing drug designing utilizing AI, I know Utsavji is here. In his family, people are there who are developing drugs through the AI implementation. Also the paperwork and the trial design, and then all of that things have been rapidly advanced. AI has also opened up the knowledge of the access of knowledge quite rapidly. It has also increased, what I would say, that bubble has started where the questions and curiousness was solved in research conferences once in a year. And now I personally see that some queries can be answered very quickly by AI to a scientist. So I think it is rapidly evolving, and I see a lot of potential use of it in biomedicine.
So we are
Thank
running
you.
short of time. We will take two more questions. First one is from Dr. Vineet Singh from Canada, and second one will be Dr. Siva Raman. Dr. Vineet Singh, do you want to come on camera and ask a question directly to Dr. Tej Pareek? No response. So in that case, we'll ask Dr. Siva Raman to ask his question.
Yeah, so I know it's too early to go into safety and all of that, but especially with the COVID mRNA vaccines, people had seen a lot of things like myocarditis and uh, stroke and clotting and stuff like that. Now, that might be less of an issue when somebody is fighting for their life with cancer. But what do you see there? I mean, do you think that could be a problem with mRNA vaccines for cancer?
There is always something, anything which is done artificially to our body has a chance to tell us something else, and nature doesn't reveal its secrets so easily.
Hmm.
So humans think that we have found the cure, but when it goes into body, there are reactions, and then body respond to it. So no scientist, no doctor will ever give you the answer where you will
Right.
say that everything is super safe, this is the magic bullet, and you take it. uh, Things will happen, but that's why we have these clinical trials where we go into larger population. We also do risk benefit analysis, that what is the disease we are curing, and a some small incidents will be okay to take on that. However, I can tell you, Shiv Kumaranji, like these, even the dendritic cell vaccines, there are a lot of clinical trials with different approach has been. And theoretically, whatever understanding of science we have today, they are much, much, much safer than the traditional chemoradiotherapy approach, which we know. So our basic common sense knowledge suggests that they should be safer. However, once we will be testing in this larger population, then only we will know. And that's why we do the step by studies where we first invest 5-10 years just to do the safety trials, to make sure that forget about cancer will be cured or not, to make sure there will be no adverse effect of of any intervention which we are doing. So I think I cannot give you the exact answer. It will be safe, but that's how science is driven.
Yeah. Thank you.
Well, thank you very much indeed, Dr. Tej Pareek, for giving a very fascinating lecture. I see very good feedback actually from the audience members. You made a very complex topic so easy to understand in simple language and terminology. So, thanks a lot for that. We wish you success in your venture and hope that the clinical trials, are initiated phase 1, phase 2, phase 3, because cancer is an area which is kind of increasing in terms of inception rates. And as we are living longer in a chemicalized environment, there are more triggers to initiate new types of cancer. So any research there is going to be protective to this species that we have on the earth. Thank you very much indeed to our audience members. And finally, thank you to Dr. Siva, who is, our technical support today. So thanks to everyone.